Hello, everyone. First, I'd like to thank the organizer for inviting me and being interested in this work. It's a true honor to be here with you today. The title of my talk, The Death of Short Noise, Removing Uncorrelated Noise in System Neuroscience, is intentionally provocative. And the reason for this is my view on designing two-photon instruments and using the data has completely changed through this work in the last few years. And so I would like to share this with you and potentially discuss and sort of so that you can bring this insight with you home. So let me start with an example movie. So this is a GCamp a movie recording GCamp activity in neurons in the visual cortex at the Allen Institute. So this was released to the public. You see here individual somas of neurons, but when I play this 30 Hertz movie, I think the most striking features of this is the presence of speckle throughout the whole field of view in this raw movie data. And this is essentially short noise. Indeed, in low light regime, as is often typical in system neuroscience recordings, we are dominated by essentially the quantization of light. We don't have enough time to average out those events, and as a result, our SNR actually scale the square root of the number of photons. And this has impact on all downstream subsequent steps. In practice, all microscopy developers know about this limit and the design around it. So in this graph, Natalia, Orlova, and Benjamin Grew and myself, we did this figure for a review in Neuroscience a couple of years ago. And what we did is we took a number of two-photon technical paper introducing new techniques. And then we looked at the number of neurons that were sort of recorded in those experiments, along with their frame rate. And sort of the trade-off imposed by short noise is apparent in this graph. Essentially, time is your currency in this case. You can spend time recording an individual neurons at very high frame rate, as shown here with U-log, or you can sort of trade off the speed for more neurons, as is shown here with Trepan 2P. Ideally, where we would like to be is here, a larger number of neurons with high frame rate. But we can't reach that point because of the limit of the amount of signal we can record, and essentially because of short noise. But we want a limit. One way to get there, though, is by gaining time. And one way to gain time is to have more lasers scanning the tissue. This is what we did with Oleg Rumyantsev and Mark Schnitzer a couple of years ago for a recent paper. In this scheme, we used a 16 laser beam arranged in an array pattern in a checkerboard, and smart spatial-temporal multiplexing paired with very sophisticated mechanical and optical engineering done over a couple of years to have this sort of 2 by 2 millimeter field of view scanned with this 16 laser beam, and each beam was associated with a dedicated photodetector. And this allows us to scan a couple thousand neurons with a 10 Hertz frame rate with dendritic resolution across the whole field of view. This approach, you could think about, this approach could scale up. You could have even more lasers and then record the entire brain. But obviously, you fall into the limit of how much the brain can take in terms of light excitation. You will reach photo damage, essentially. In this study, we're careful to avoid this threshold, but this could become a problem if you want to scale this up further. And so I was trying to find solution around this problem. And before we get into this, I'd like to sort of preamble this with a little bit of insight about how much information we have. If you take an individual pixel in a two-photon movies and then try to extract the amount of information between that pixel and its neighbors. This is what I did here. I measure essentially the mutual information between a pixel and its neighbors. You can do this in space or in time. At 0, what essentially you're looking at here is the entropy, is the information you need to reconstruct that pixel. you will see that there's a fair amount of information in its neighborhood. What that means is that if we were to extract all the information that is present nearby pixel, we could actually reconstruct the value of the center pixels. But what would that would do is, because there are many more neighbor pixels that share information with center pixels, that would effectively greatly increase our photon bandwidth by probably orders of magnitude, thereby eliminating the underlying limit at the core of short noise. The reason for short noise is because we don't have enough photons to measure weak fluorescence values. Now the question is, how can we learn this complex nonlinear spatial-temporal relationship? The problem is in system neuroscience, we don't have sort of ground truth data. And the traditional approach will be to have like a deep learning network with ground truth data showing the actual real value of the pixels, and learn this relationship between pixels to train a reconstruction network. But because we don't have a ground truth data, this is made difficult. A couple of years ago, this paper came out that just literally blew my mind, and I'm sure it influenced a lot of people in the field. This is a sort of a basic deep learning research paper, and they call their approach noise to noise. It was published by a group at NVIDIA under the leadership of Lehtinen. So their idea was, as I said, the traditional approach is to have clean data and to map a noisy data to clean data to train a denoiser. In the case of, let's say now that you have multiple instances of that image, so the same image, but with different types of noise on top of each other, on top of it. If that noise fulfills certain condition, an interesting phenomenon happens. You can train a network to map one instance of that image on another instance, and because the noise means zero, the reconstruction loss that you will use on that image will, on average, point the gradient towards the right direction. So what happens during training is that the image will gradually become like the clean image, even though the network had never seen this clean image. And we can actually use this insight in two-photon microscopy with a couple of small modifications. Now, we don't have multiple versions of the same image, the ground truth image, with different instances of noise in two-photon calcium imaging, but we do have a long series of image, and they share a relationship with one another, as I shown in the previous slides. So what we can do is take that center image that we want to predict and place it on the output as the original noise-to-noise implementation, and use all nearby image to try to predict it. Now, we're not sort of doing a one-to-one image prediction. We are drawing an interpolation now, thereby the name DeepInterpolation. And because the short noise fulfills these properties of being mean zero, in the end, we're going to converge to the right solution. So even though the network had never seen the true clean image, it will train and converge to the right solution. And this is what happens here. During training, the validation loss decreases and converge to the noise floor. It cannot go to zero because short noise dominate. During inference, you will take that network back after training and just stream it the movie through it and end up with like a denoise data on the output. This approach is similar to two other papers called noise to voice and noise to self, although they applied it to single frame denoising, whereas here we are training complex spatial-temporal models using large databases. This is how it looks in practice. You take a two-photon movies, and you, as I said, you omit that image, and you place it on the output. I use a typical unit architecture with skip connections, and as through training, you will see that it will converge to the true underlying image. This is the sort of performance during training, and so what we did is we used the large database present at the Allen Institute to train this network. What we wanted to do was to have a fairly good general purpose denoiser. And because we had literally thousands of 1-hour recordings, we had billions of frames to train it. And so we sample randomly a subset of frame from many hundreds of movies to train this denoising network. We found that the larger the input range, so the more frames were provided in the input, the better the validation loss during training, and that after as little as 200 to 400,000 frames, the network had sort of reached stable performance. And you see here in the raw frame, the raw frame compared to 25,000 frame and 400,000 frames of training, the definition of the image is improving, and now you start to see much smaller compartment and very better defined. Now, if you take and you extract individual somatic ROI, region of interest from that movie, and look at the calcium trace, the calcium traces are preserved, and the sort of the denoise trace is following faithfully the underlying noisy signal. This is true both at the somatic level and importantly at the single pixel level. If you take the SNR of all of the pixels in that movie, you will see that essentially the SNR is multiplied on average by 15, which is a huge gain in signal quality. On similar par to going from GCAM5 to GCAM6, essentially. This is an example of this. So we trained the network on using the large database at the Allen Institute and applied on LDAT dataset from the Allen Institute. And so I wanted you to look at two things. So this is the raw, and on the second column, we subtracted the mean so that you could see more clearly the difference. And the bottom is after deep interpolation. So you will immediately notice that the denoise movie is much better defined. You can see smaller compartment much clearly. Sometimes there's actually instances of spines here that are visible, which was hidden in the noise before. And also, you should notice that the raw movie had leftover motion artifacts, a little bit of jitter present in it, which were actually entirely corrected after denoising, because this was treated as a little bit of independent noise at the single frame level. This also works across a range of movies. So this is the same denoising network applied on 4 different LDOT movies, and so of many different sort of SNR conditions. And the sort of the motion correction properties is a little more apparent, for instance, on this movie. But in all cases, the reconstruction was quite faithful. Okay, so you might ask, okay, it looks great, but how faithful is this compared to ground truth data? So obviously the problem is we don't have a ground truth in system neuroscience, but we can find a workaround. So what we did is we took simulation data created with Naomi. Naomi is a simulator created by David Tank's group, by 2 of his former employees, Charles and Songs, which is now is a PI, and Song was a PhD student. And the beauty of this approach is that it leveraged fake geometries, so fake somatic geometries and dendritic geometries, and inject it with calcium dynamics and have a sort of an artificial laser beam scanning the tissue. And so it's recreating very faithfully how real data looks. And it's actually in practice, looks fairly convincing, to the point that I think many experimentalists could be confused which one is the true data set here. So we use this data set where we had sort of simulated noise as well as ground truth to compare deep interpolation with other approaches that are present in the field. This is what we did here. So this is the simulated data with ground truth in orange and the noisy version of the ground truth in blue. And we did the Fourier transform here so that you could see that indeed short noise is injecting higher frequency noise. Now, we compare this with penalized matrix decomposition, which is an algorithm developed by Liam Pandinsky lab, which actually performed quite well in a single frame. But when we look closely, we found that the main difference is with the sort of the higher frequency projection of the algorithm. But indeed, a lot of the high frequency noise was still present after PMD. And some of the spikes were also sometimes left over. And we found that when we compare this with deep interpolation, deep interpolation was much more faithful to the original sort of distribution of frequencies, and also the L2 norms, so that the reconstruction quality of the ground truth signal. Deep interpolation compares well with other techniques. We actually also compare with Gaussian kernel. And the Gaussian kernel was good in noise rejection, but introduced a lot of sort of artifactual smoothing of the trace, as shown here. If you zoom in on an individual event coming from a single cell here, you see that deep interpolation is faithfully following the calcium dynamic, which is a good thing to see. All right, what's the impact now on subsequent step like segmentation, say, for instance? So what we did here is we took Suite 2P developed by Marius Pashitario and Carsten Springer and apply it on raw and data after deep interpolation. And what we found is we did more quantification, but this is just for illustration purposes. We found that the cells that were higher SNR look very comparable before and after deep interpolation. But as you dive deeper in the data, you will see that the cell with sort of more noise in them, lower SNR, where had less clearly defined region of interest, and sometimes we're missing part of the soma, but we'll recover after deep interpolation. Sometimes even we'll sort of see a small neuronal segment, dendrite, where in the region of interest automatically detected by Suite 2P after denoising. And to quantify this further, we looked at now not comparing cell match cell before and after denoising, but looking at only at the output after deep interpolation. And interestingly, we found that a large number of new segments were found. So we saw somas as before, but we also saw a lot of horizontal dendrites that were detected now in the denoise data, as well as small vertical segments. So like a compartment that are going through the field of view vertically. So they could be either dendrites or axons. And remarkably, all of them had very clean calcium signal. We quantified the SNR of all of this. And so what happened is essentially denoising, such as just leveled the fields across all compartments, regardless of their size. Indeed, if you quantify the SNR after a deep interpolation, all region of interest had an SNR above 20. So to give you a good sense of perspective, if you want to detect a 10% event, you need the SNR to be above 20. And this had an impact on the biology. So now if you look at the response to natural movies, so the mouse was stimulated with visual stimuli, the algorithm had no knowledge about that, but we quantified the response reliability of individual region of interest to the visual stimulus. And we found that all of them had increased response reliability to this external stimulus after deep interpolation. So now you might ask, okay, it sounds great. You were able to train these 15 million parameters with Allen Institute dataset. Can we use this tool for my own data where I have maybe less dataset to train this on? And so to answer that question, what we did is we took different imaging conditions from the condition that network was trained on. So let's say we went to a sort of changing the rig or going from excitatory to inhibitory neurons, we're not present in the training data set, to layer 6, which is lower SNR conditions, or a complete new imaging protocol going from 30 to 10 hertz recording. And we did another round of training to see whether fine-tuning network could recover the reconstruction performance. And surprisingly, we found, and this has been shown in the deep learning field, we found that as little as 10,000 frame was enough to sort of reach stable validation loss values, which meant that, keep in mind that a single 1 hour recording in system neuroscience is as much as 100,000 frames. So we could fine tune this network just with a single recording. And I have an example of this sort of reconstruction performance in this example here. So this is raw data. This is a layer 6, so very dim and noisy data set. In the middle, this is before transfer training. So we actually took the raw network trained on the Allen Institute database and apply it to layer 6 that I was never trained to it before. And after after fine tuning or transfer training on the third column. And here again, I'm showing the mean subtracted version of this. And so I want to sort of look at the background difference. So you see that before transfer training, there's a lot more sort of background fluctuations. The network is not performing perfectly well. The cells are still active and reconstructed. It's a big improvement from the raw data, but there's still this fluctuation in the background, which are totally gone after you fine tune the network and sort of force it to match the data set that it needs to work on. So I'm confident that, and some collaborators of us have actually confirmed this result using data set from completely outside the institute, that the minimal amount of training is sufficient to reconstruct a reconstruction performance with our pre-trained network. Importantly, indeed, all the data is available. It's on GitHub. We sort of released all the code, including all the code to regenerate the figures here. The pre-trained network on Dropbox, so you can actually take it. There's a support channel on Slack, so feel free to ping us and we will help you. And there's also like a TensorFlow model inference on Docker Hub, so if you want to deploy this on AWS. So to conclude, I hope I've shown you that we've developed a deep interpolation algorithm that does not need ground truth. It could be applied to a large family of spatial temporal data sets. So I believe this is a general purpose algorithm that could be used in microscopy as a whole, and neuroscience a lot of these. More data yields improvement in noisy model. So this can leverage large databases of standardized data set, as we've shown. And so I think this is a testament to sort of a big data approach in neuroscience. I've shown you that fine-tuning pre-trained model with minimal amount of data set size can recover the reconstruction performance in neuroimaging conditions. And it's entirely self-supervised, so no domain expertise is required except the deep learning expertise. Reconstructing two-photon movie without shot noise has a large impact on all subsequent steps. We've shown cell segmentation, we've shown motion correction. And I believe that combining this insight, this denoising approach with efficient laser scanning methods could allow an order of magnitude increase in the number of recorded neurons, because we need less sort of photons now that we can leverage the relationship between pixels. And so I'm hoping that this might be a component that enable large-scale voltage imaging in the future. And I would like to thank my collaborators, Michael Oliver, Josh Siegel, Christophe, and Natalia, were tremendous help in getting this to work, but it could not work without them, as well as the NIH funding of this work, as well as the Allen Institute. And I thank Paul Allen for his vision, encouragement, and support, which was tremendous in setting up the Allen Institute. Thank you.